friends, now we are going to see about the topic of neural control and coordination based on NCERT. Coming to the first, we have to know about coordination. What is coordination? Priorly going into the coordination, I will tell you a very simple example about the group dancing. I think we all know in the group dancing, many people will be dancing in a synchronized fashion. For example, if anyone was not in that synchronized fashion, it will leaves the daisy form of the dance. Means nothing but in coordination. So that is the one a typical example about the coordination. Likewise, in our body also, for example, walking is a coordinated movement. Hands, legs, body, everything has to be work for the walking. If anything was being disformed or any disproportionate, then it leads to incoordination walking. So this is the best example for coordination. I hope you got it. First, then we will go to the whole process of coordination now. So the coordination is the process through which two or more organs interact and complement the functions to one another. For example, when we do a physical exercise, all body parts will coordinate. Neural system and the endocrine system jointly coordinate and integrate all the activities of the organs so that their function is a synchronized fashion. Coming to the next, so this is a simple flow chart about the neural system. Starting for the first, in this video we are going in a simple manner. Later in the other videos we are going to discuss in a detailed perception. Right. Coming to the neural system, we basically divide the whole neural system into central nervous system and peripheral nervous system. Nervous are neural, both are similar only. Coming to the central nervous system, we have the brain and spinal cord. Coming to the peripheral nervous system, we have the somatic nervous system, autonomic nervous system. Somatic means under our control, voluntary actions. Autonomic nervous system means which is automatic in nature. For example, heartbeat and digestion, all these things comes under autonomic neural system. And again this autonomic neural system is being divided into sympathetic neural system and parasympathetic neural system. I have discussed about this topic in the previous video. Please do and check. So sympathetic nervous system we also call it as flight or fight response. Parasympathetic nervous system we also call it as rest and digest nervous system. And coming to the next, the neural system of all animals is composed of highly specialized cells which we call it as neurons, which is the basic structural and functional unit of the nervous system, which can detect, receive and transmit different kinds of stimuli. So the neural organization is very simple in the lower invertebrates. For example, in Hydra, it is composed of a network of neurons. The neural system is better organized in insects, where a brain is present along with a number of ganglia and the neural tissue. So the vertebrates have a more developed neural system. So the neural system is being divided into two parts. We have already seen in the previous slide, central neural system and peripheral neural system. The CNS includes brain and spinal cord. The PNS comprise all the nerves of the body associated with the CNS. Anything coming out of the CNS we call it as PNS. Nerve fibers of the PNS are of two types, afferent fibers and efferent fibers. Afferent fibers are those the nerves send stimulus transfer message from the sensory neurons to PNS. For example, if we touch anything, we can feel the texture of the skin or texture of the cloth because of this afferent fibers. Means it will take the information from the receptor towards the brain. Next, efferent fibers. So efferent fibers are those neurons take the message to motor neurons. For example, if you are touching any hot thing is being detected by afferent neurons and the hand should be taken immediately from the hot pan. So that has to be decided by efferent fibers because it receives the messages from the brain through motor neurons. So the PNS is being divided into two divisions, somatic neural system and autonomic neural system. Somatic neural system relay the impulses from the CNS to skeletal muscles. Autonomic neural system transmit the impulses from the CNS to involuntary organs and smooth muscles of the body. So the autonomic nervous system is further classified into sympathetic nervous system and parasympathetic nervous system. Coming to the neuron as structural and functional unit of the neural system. This is the diagram of the neuron. Now we are going to discuss about this diagram. So initially we have the dendrites, this tree-like projections we call it as dendrites, and the center spherical type of organelle which we call it as cell body or perikaryon. And one more interesting thing about the neurons are the neurons are the longest cells in the body. And this perikaryon, inside this perikaryon, centrally located nucleus, we have the mitochondria. And from here, there is an elongated, stretched structure, which we call it as axon. So coming to the axon, on the axon, we have a myelin sheet, so which acts as an insulator. This myelin sheet is being produced by Schwann cells in the peripheral nervous system and oligodendrocytes in the central nervous system. This is very, very important point. And we have the nodes of Ranvier. Where the myelination is not present, we call it as nodes of Ranvier. The distance between node to node, we call it as internode. And these are the Schwann cells. And we have the nerve terminals, so axon terminals. Axon terminals will connect with the other neurons by synapse. So that junction, chemical junction, which we call it as synapse. And coming to the next, we have to know certain definitions of these terminologies or vocabulary based on neural system. Neuron is a microscopic structure and functional unit of nervous system composed of three major parts, namely cell body, dendrites and axon, as we seen in the picture. Nissl granules. The cell body contains cytoplasm with the typical cell organelles are certain granular bodies, which is nothing but Nissl granules. Nissl granules are nothing but typically ribosomes. These are, we also call it as trigoid bodies because those are triangle in nature. So that is very important. So it is been the characteristic feature of neurons. You can only find the Nissl granules in neurons. No other cell will contain the Nissl granules. That is the characteristic feature we have to remind. Next, dendrites. These are the short fibers which branched repeatedly and projected out of the cell body. So these fibers will transmit the impulses towards the cell body. Right. Next, axon. It is an elongated fiber of the cell body. End is being branched. So its end is branched. We will go back again. So these are the dendrites which will bring the information towards the cell body. And from the cell body, the information is being transmitted to axon. So which is again the end is being branched. Finally, it will end up with axon terminal. From the axon terminal to it will go to the other neuron through synapse. And next, synaptic knob. At the end of the branched axon has a bulb-like structure which we call it as synaptic knob. Next, synaptic vesicles contains the chemicals called neurotransmitters. Neurotransmitters is chemical which take part in the transfer of messages to the other neurons. The neurons are having three types. We have three types of neurons based on their polarity. Coming to the first, multipolar with one axon, two or more dendrites found in the cerebral cortex, which we call it as multipolar neurons. So in this picture, what we have seen, this is multipolar neuron. Nothing but we have many poles, 1, 2, 3, 4, 5, 6. We have the many poles. Because of that, we call it as multipolar neurons. And bipolar neurons with one axon and one dendrite found in the retina of the eye. So this is found only in the retina of the eye. That is the characteristic feature. Again, next, unipolar neurons. So it is generally in the matured body, no unipolar neurons will be there. So where it is present means during embryonic stage, like a sprouting from the neurons. I think, but during the development of the neurons, you can find the unipolar neurons. So unipolar, the cell body with one axon, only found usually in the embryonic stage. Next, there are two types of axons, namely myelinated and non-myelinated. So myelinated nerve fibers are found in spinal cord and cranial nerves because myelinated nerve fibers will work very fastly than the unmyelinated neurons. So in order to take the message to and fro, we need the myelination. Because of that only they are present in the spinal and cranial nerves. The myelinated nerve fibers are enveloped with Schwann cells, which will form a myelin sheet around the axon. The gaps between the two adjacent myelin sheets are called nodes of Ranvier. Next, unmyelinated nerve fibers is enclosed by Schwann cells and that does not form a myelin sheet around the axon and commonly found in autonomous and the somatic neural system. Next, generation and conduction of nerve impulse. The neurons are excitable cells because their membranes are in the polarized state. Polarity means inside it is negatively charged, outside it is positively charged. So it contains the polarity like the batteries. So because of that, it is polarized state. Different types of ion channels are present on the neural membrane. These ion channels are selectively permeable to different ions. For example, if those are sodium channels, means it will allow only the sodium. If it is the potassium channels, means it will allow only the potassium. Likewise, based on the ions, they have a different channels. Next, there are two types of state potential. One is resting potential, other one is action potential. If you are in the rest position, all the potential is being takes place is resting potential. So the electric potential difference across the resting plasma membrane, we call it as resting potential. Inside it will be negative and outside it will be positive. That we call it as resting membrane potential. When we are excited and active, when the impulses is being sending to and fro, then we call it as action potential, means the nerve is in action. So the electrical potential differences across the plasma membrane at a site A, which we call it as action potential. Next, transmission of impulses. So this is the diagram which will depicts about the transmission of impulses from dendrites to cell body to axon, axon to the axon terminal to synapse, which is a chemical border between this, and from the synapse to the other dendrites, cell body, and again to the axon. So the transmission of impulses from one neuron to the other neuron will take place in this unidirectional fashion. And next, so in this picture it is clear about the how is the axon terminal and synapse. Coming to this picture, so in detail if we want to discuss from this picture itself, starting from, so this we call it as presynaptic membrane and this we call it as postsynaptic membrane. So this whole thing is synapse, right? So presynaptic membrane and postsynaptic membrane. In between this space we call it as synaptic cleft. So these three parts are very important. First we are going into this. And now we are coming into the axon end bulb. So in the axon end bulb, we have vesicles. So these are the vesicles. What inside this vesicles is been consists of neurotransmitter. For example, we have many neurotransmitter like acetylcholine, GABA, serotonin. So these are the examples for the neurotransmitters. So for example, you assume we have the acetylcholine, ACH. So acetylcholine is being presented in a form of packed vesicles. So, and now when the axon terminal is reaching till the presynaptic membrane, the vesicles will start moving towards the end. When it comes to the end, through exocytosis, nothing but giving the contents away from the cell, through exocytosis, it will release the ACH into the synaptic cleft. Now, the ACH will get attached to the postsynaptic membrane. On the ACH receptors, not it will go inside this membrane. So, this ACH is being attached to ACH receptors, and now again from here the action potential will start. So this is the pictorial representation of synapse. Next, transmission of impulses. A nerve impulse is transmitted from one neuron to another neuron through junctions called synapses. So you please check whether you are understood or not from this description. A synapse is being formed by a membrane of presynaptic neuron and postsynaptic neuron, which may or may not be separated by a gap called synaptic cleft. There are two types of synapses. One is electrical synapses, other one is chemical synapses. The electrical synapses are nothing but the membranes of pre and postsynaptic neuron and in very close proximity. It is rare in our system. It is not present in humans. So electrical current can flow directly from one neuron into the other across the synapses. So the impulse conduction along a single axon, and impulses of electrical synapses is always faster than the chemical synapse. Mostly you can see these electrical synapses in fishes. Because of that, it will act in a very fraction of second because of this electrical synapses only. Next, chemical synapses. The membrane of the presynaptic and postsynaptic neurons are separated by a fluid-filled space, which we call it as synaptic cleft. Next, neurotransmitters are involved in the transmission of impulses at the synapse. So the synaptic vesicles filled with neurotransmitters, we have been discussed in the picture in the before slide. So when an impulse or an action potential arrives at the axon terminal, it stimulates. So the movement of the synaptic vesicles towards the membrane, where they fuse with the plasma membrane and release their neurotransmitter in the synaptic cleft. So the release of the neurotransmitter bind to a specific receptors. So the binding opens the ion channels, allowing the entry of the ions, which can generate a new potential development, maybe either excitatory or inhibitory. All the times we will not be excitatory. Sometimes we will sleep also. Because of that nature, based on the nature of the stimulus, it will be excited or inhibited. Next, nervous system. The brain is the central information processing organ in our body, and it acts as the command and control system. It controls the voluntary movements like balance of the body, functioning of vital involuntary organs, for example, lungs, kidney, heart, etc. The brain is protected by skull and covered by cranial meninges. So, the cranial meninges have three layers: dura mater, arachnoid matter, and pia mater. Dura mater, so the name itself, so to find out it very easily, dura, which comes from the word durable, which is the very thickest outer layer of cranial meninges. And next, arachnoid matter. Arachnoid means spider web. It looks like a spider web between the dura mater and arachnoid matter. So, that is the middle layer. Next, pia mater. Pia mater is the very inner layer that will directly contact with the brain tissue. So whatever the gyri and sulci, along with that the pia mater also will flow. So in order to remind this very easily, you just remind PAD, pad, right? So pad, from inside to outside, PAD, pad. And next, in this picture we can able to see the forebrain, midbrain and hindbrain, right? So coming to the brain, so we have the cerebrum. So cerebrum, this cerebral hemispheres, all these things will comes under forebrain, means front. And this is the midbrain. And we have the hindbrain. So we have the cerebellum, pons, medulla oblongata will comes under the hindbrain. Pons, cerebellum, medulla oblongata. And we have one more terminology which we call it as brainstem. Brainstem is nothing but midbrain, pons, medulla oblongata. Midbrain, pons, medulla. So this looks like a stem. Because of that, this has given the terminology brainstem. So this is corpus callosum, which will connect both the cerebral hemispheres. And this is cerebral aqueduct. Aqueduct means passage. So these are the ventricles. What we can see here is the ventricles. Cerebral aqueduct. We have the pons, which is the bridge, which is the meaning, exactly the meaning of pons is bridge, which acts as a bridge between midbrain and medulla oblongata. And we have the cerebellum, which is the little brain, which is for the balancing of the body. And next, medulla oblongata, the major respiration, heartbeat, everything is being controlled in medulla oblongata. If anything happened to this medulla oblongata, the person will die immediately. And we have the thalamus, which is a sensory relay station, and hypothalamus, which is glandular in nature. So all these parts are present in this picture. And coming to the forebrain, they have three major parts: cerebrum, thalamus, hypothalamus. The cerebrum longitudinally is being divided into two halves, which is being termed it as left and right cerebral hemispheres. They are connected by a tract of nerve fibers, which we call it as corpus callosum. The layer of the cells which covers the cerebral hemisphere, we call it as cerebral cortex. We have seen in the previous chapters also, cortex means outer side, medulla means inside. So the cerebral cortex is being referred to as grey matter. Due to the greyish appearance, due to concentration of neurons, it contains motor, sensory and the larger areas. The region which we also known as association areas are responsible for complex function like intersensory associations, memory and communications. Fibers of the tract are covered with a myelin sheath, the inner part of the cerebral hemisphere, an opaque white appearance layer, which we call it as white matter. If wherever in the neurons, if it is white in nature, means it is being covered by myelin sheath. So typically, compositionally, myelin sheath is nothing but fat. Because of that, it looks white in color. And the cerebrum wraps around the structures which we call it as thalamus, sensory relay station, which is a major coordinating center for sensory and motor signaling. Next, hypothalamus, which lies at the base of the thalamus. It contains a number of centers which controls the body temperature, urge for eating and drinking. It also contains several groups of neurosecretory cells which secretes the hormone called hypothalamic hormones. The hypothalamus, it is involved in the regulation of sexual behavior and expression of emotional reactions. Next, midbrain. Midbrain is located between the thalamus and hypothalamus. A canal called the cerebral aqueduct passes through the midbrain. So the dorsal portion of the midbrain consists of four rounded swellings. Dorsal means backside of the midbrain consists of four swellings, which we call it as corpora quadrigemina. So it is very easy to remain. Corpora, corpus means body, quadrigemina, it looks like four circles. So because of that, they called it as corpora quadrigemina. Midbrain and hindbrain will form the brainstem. Next, hindbrain. Pons consist of a fibers tract that interconnect the different regions of the brain. So cerebellum has a very convoluted surface. Convoluted means many up and downs. They provide an extra space for many neurons. Medulla of the brain is connected to the spinal cord. It controls the involuntary actions. Next, reflex action and the reflex arc. So in this diagram, we can go to depict about. So we have this muscle spindle receptors. These are the afferent pathway. So the afferent pathway will takes the information from the peripheral neurons to central nervous system. Nothing but, for example, in this picture, it is spinal cord. So from the muscle spindle receptor, the afferent pathway will take the information, and this we call it as dorsal root ganglia. So means this is the ganglia which is present outside the central nervous system. Nothing but concentrated nerve cell bodies. So from here it will go to spinal cord. From here, so into the white matter. From here, these are the efferent pathway. So this efferent pathway will go to many neurons. Here they have been shown to, it is going to two different muscles. So the information is being again taken through the motor neurons. So afferent is sensory, efferent is motor. So showing the knee jerk reflex. We can also see if we hit on the kneecap, immediately without involuntary our knee will flip. So this we call it as knee jerk reflex. So this is the knee hammer. When they are hitting the muscle receptors, is been taking the information through afferent pathway to the spinal cord through the dorsal root ganglion. From here, so this is the center. From the center through the interneuron. So this is the interneuron, which is a connection between the afferent and efferent. From here, the impulse is being through effector neuron or the efferent neuron. The information is being taken to the muscles to react, means to flip, which is an action. So this is the diagrammatic representation of reflex action. Coming to the reflex action and the reflex arc, the entire process of a response to a peripheral nervous stimulation that occurs involuntarily without requiring the involvement of the part of central nervous system, we call it as reflex action. For example, if has to involvement of the brain, it takes a huge amount of time to go to spinal cord, and from there to brain, and again from brain to spinal cord, and spinal cord to the effector. All these things, it takes a large time, but in the reflex action, a very short period of time is being covered. The reflex pathway comprises at least one afferent neuron and one efferent neuron, approximately arranged in a series, which we call it as reflex arc. So coming to this cross section of the eye. So here we have the cornea. So this is the cornea, the very outer layer. And the outer layer to this cornea, we have one more layer which we call it as conjunctive. And this is the iris muscle. And this is the lens. And this is the vitreous body. And here we have the, so whole, it is been divided into choroid layer, and we have the macula. So this we also call it as blind spot, macula. Retina, very inner layer is the retina. The very outer layer is the corneal layer, sclera. Sclera, the very outer layer is sclera, and the middle layer is choroid layer, and very inner layer is retina. So in the retina only, all the neurons is being concentrated, and this is the optic nerve which goes to the brain. Coming to the eye, our paired eyes are located in the sockets of the skull, which we call it as orbits. The wall of the eyeball has three layers. One is sclera. The white color layer, what we see is nothing but sclera. Is the external layer is being composed of dense connective tissue. Choroid, in the middle layer, which contains many blood vessels and looks bluish in color. If we cut this white layer, you will get a bluish layer, which is a choroid. It becomes thick in the anterior part to form a ciliary body. So this is the ciliary body. So this is the ciliary body. From the ciliary body only, the iris muscle is being developed. So ciliary body, cornea, is the very anterior portion of this layer, which we call it as cornea. Next, the ciliary body continues forward to form a pigmented and opaque structure, which we call it as iris. Iris means Greek goddesses of rainbow. Because of that, the name iris has been came. So the infront aperture is being surrounded by iris, which we call it as pupil. Pupil is nothing but that hole inside our eye. For example, when we go into the movie theater, that pupil will become very large, means the circle will become very large. When we go into a flashlight or a floodlight, that circle will become very less. So that we call it as pupil. So the eyeball contains a transparent crystalline lens, which is a biconvex lens. The inner layer of the retina, and it contains the three layers of the cells from inside to outside. Ganglionic cell, bipolar cells, which have seen the types of neurons. Ganglionic cells, bipolar cells, photoreceptor cells. We have to remind this order. This is very important. There are two types of photoreceptors, namely rods and cones. So cones is for bright light vision, rods is for dim light vision. You always remind cone for more light accommodation, rod is for less light accommodation. So you can remind in that manner also. Optic nerves transmit the signals to the visual cortex of the brain to know the image. So ear. So this is the diagrammatic representation of the ear. If you want to know much detail about the ear, I have done a video on the ear and whole anatomy and mechanism. Please refer to that video. It will be somewhat interesting to know. As usual, it is more descriptive in nature. And to know the parts here, this is the ear pinna. So the whole ear is been divided into three parts. External ear, from here to here it will be external ear. In the external ear we have pinna, external auditory canal, tympanic membrane. Till the tympanic membrane we call it as external ear. And this part, this pinkish part which is being covered, this we call it as middle ear. In the middle ear we have ear ossicles, nothing but malleus, incus and stapes, which is the smallest bone in the human body. And this is the eustachian canal, which connects to the nose, middle ear to the nose is being nasopharynx is being covered by this eustachian tube. So this is the middle ear, and this whole vestibular apparatus, cochlea, everything which comes under in internal ear. So we have external ear, middle ear and internal ear. So this is the snail like structure which we call it as cochlea. So this is the vestibular apparatus. So this is for equilibrium. This part is for equilibrium and this part is for hearing mechanism. So we have the vestibulocochlear nerve, eighth cranial nerve. So the cochlear is for hearing, vestibular nerve is for balancing. And ear has been divided into three parts. Outer ear consists of pinna and external auditory meatus or canal. The external auditory meatus extends up to the tympanic membrane or the eardrum. There are very fine hair and wax secreting sebaceous glands in the ear canal. So ear wax, nothing but is being secreted by these sebaceous glands. The middle ear has three ossicles. So one another is in a chain-like fashion. Malleus, it is being attached to the tympanic membrane. Incus helps in the vibrating effect. Stapes is attached to the oval window of the cochlea. So malleus attached to the tympanic membrane. So in between we have the incus, which is being attached to the malleus and stapes. And the stapes foot is being attached to round window. Here it will be the round window. Oval window. It is oval window. Next, eustachian tube. It connects the middle ear cavity with the pharynx and equalizing the pressure in the ear. Next, the fluid filled inner ear, which we call it as labyrinth, consists of two parts, the bony labyrinth and membranous labyrinth. The coiled portion of the labyrinth, which we call it as cochlea. The space within the cochlea called scala media is being filled with endolymph at the base of the cochlea, and the scala vestibuli will ends at oval window. How do we hear the sound? The organ of Corti is a structure located in the basilar membrane which contains the hair cells that acts as a auditory receptors. So a large number of processes, which we call it as stereocilia, are projected from the apical part of the each hair cell. Above the rows of the hair cells is a thin elastic membrane which we call it as tectorial membrane. The inner ear also contain a complex system which we call it as vestibular apparatus, located just above the cochlea. So the vestibular apparatus is being composed of three semicircular canals. We can very easily remind this. We have anterior semicircular canal. For example, if you move the head from back to front, then it is anterior semicircular canal will be in this direction. If you move your head from side to side, so if you bend your head from side to the side, that is posterior semicircular canal. If you rotate your head horizontally, that is the lateral semicircular canal. So we have the three semicircular canals in this direction of the head movement. And the otolith organ, so consists of saccule and utricle. So each semicircular canal lies in a different plane, means nothing but right angles to one another. So that vestibular apparatus helps for the maintenance of balance of the body posture. So thank you very much. Please do like, share, comment and subscribe to my channel for more biology videos. Thank you.